Hi, welcome to the second part of the protein interactions and docking lecture and 3D function prediction. Last time I said this is all around the function prediction is all around the idea that similar proteins have similar interaction partners. Therefore, using this similarity, we can try to find out something about the protein function. Okay. I told you this was what we're going to talk about. Last time we came until the point of binding site analysis. This time we're going to talk about that a bit more, about how to compare binding sites, and I'm also going to tell you about how to find out whether a small molecule fits into a binding site, which is called docking. Just to repeat, I thought in order to compare two binding sites, the first prerequisite is to find how can we superimpose them in 3D. However, this is kind of a hand and egg problem, because if we know how to superimpose them, we already have an idea of what is equivalent. So in cases where the things are pretty dissimilar, it's non-trivial to find out how to superimpose. If the two proteins have a high sequence similarity or a high structural similarity, then we can use that in order to say, okay, this is equivalent, and therefore we superimpose it in that way. Okay, and I thought the really interesting things why binding site comparison where it might bring most additional information is in cases where the involved proteins do not have any homology in terms of like they're evolutionarily related. And so it's people who develop a program to do binding site comparisons look for these gems, and they look for, can we find them? And here I'm showing you one example that people looked for. These are two proteins which are completely independent from the name, which for, of course, people who don't have to do so much with biology, reads pretty complicated, but we can take it apart. So CAMP is a term for some compound that is often used in biology. Dependent protein kinase. And a kinase is always something that takes a phosphate of something and attaches it to something else. Kinase always attaches phosphate somewhere. So this is one kind of reaction, and here we have a reductase, which means this is something that adds electrons to a compound. So transferring a phosphate or adding electrons is a pretty different reaction, and therefore we can also expect that these proteins do not really have a lot in common. However, part of the stuff they use is similar, and the compounds they act on have this part in common. And because they use this compound which has a similar group, they will also have binding sites that can bind this part of this molecule, and therefore we can expect part of the binding site to indeed be similar, and that's why we think we could be able to find this kind of thing. If you look at the 2D plot of the binding site, then at first it looks like, oh no, this is completely different, I can't see anything. Now, if you turn this around a little and take out the stuff that is really dissimilar, you start to see, okay, here's the part of the ligand that is similar in both cases. And the interactions that are made are partially also similar. Like you see here, these two ends, they make hydrogen bonds to something else. And in that case, you also have this here. This end makes a hydrogen bond to that O, and this end makes a hydrogen bond to that N. So indeed, in this example, the binding site offers the same kind of interaction partners, only in this case they come from two different amino acids, whereas in this case they come, I think, from the backbone of the same amino acid. But this is the kind of thing we're looking for. We want to we want to abstract from the exact details of the amino acids, and just say, okay, we have this kind of interaction partners and they can be found. And then when you find the same pattern of interaction partners in two binding sites, then you can say, okay, probably they bind the same kind of thing. And this is also why in binding site comparison and in docking, etc., we are not so much focused on only the amino acid type. So very often in bioinformatics you just deal with the letters like A, C, whatever, and that's enough information. In this case, when you really look at chemical interactions, you often have to go a bit deeper and say, okay, really, I don't really need to see the atoms, because a lot of chemical groups can have the same kind of interaction, like an NH and an OH can both make a hydrogen bond. So you can abstract here. But you can't really just focus on the amino acid often. Okay, and so one method I'm going to show you does that by taking the amino acids that are present in the proteins and representing them by interaction sites, and then you can abstract from the exact atomic details and just see, do I have the same kind of interaction sites in both molecules? And then you can look at, do the two compounds interact in the same way with these interaction sites? Okay. So if I want to write a program to automatically do this comparison, what do I need to do? Well, first I have to be able to locate the binding site, which is what we talked about last time. Then I have to have some representation of the binding site, so I need to be able to capture the important features. And I have to have an efficient search across all the proteins. And that means I have to find the superimposition, and I need to be able to score. So I need to be able to say this match is better than that match. Okay. There are a number of methods out there. I'm going to present two of them. And as always, the presentation of what you choose is a bit arbitrary. In that case, one reason to present it is because one of the methods is, as far as I know, the first one. And it was developed in the group where I did my PhD, so I'm biased towards that one. And the other one is the new one, which is more optimized for high throughput. But if you want to learn for an exam, then I think it's always more important to get the point, why do they do it that way? What would have been other options? And so at least when I try to make exam questions, I think a lot more about what's the principle behind it than what's the detail of a certain method. Okay, so whatever method you choose, there you have a few choices for how you represent the features of a binding site. You can take the atoms. The drawback, if you take the atoms, is that you have loads. So you have a lot of different points to deal with. You can take chemical groups, and that is already some kind of abstraction, because then you say, okay, just take an OH or I take a CO or something, which is also encoding in a way the function this thing can have. Or you can completely abstract from the points where the atoms sit and say, okay, I just take the surface, because the surface is the thing that really is in contact with my ligand. And some people also have some more abstract shape descriptors. You will see what I mean a bit better when I come to the other example. Then the superimposition, there people refer to known algorithms, and I have to admit that I just take the algorithm as is and I don't really care about the details. But I hope that you will probably know a lot more about geometric hashing or graph theory or click search than I do, so I won't go into any details there because I think you'd know about it anyway. And then in the end, you can measure the similarity by just taking the RMSD. Do you all know what an RMSD is? Oh, okay. That's something we have really often in 3D structure comparison. It's the root mean squared deviation. So basically, when you, do I have more than one color? Yeah. When you have two superimposed structures, A and B, then you look for each point. How distant? What's the distance between my superimposed points? And so I have R there, R1, R2, and now I hope I won't get it wrong. So I take the square of the distances. I take the sum of the squares of the distances. I divide by the number of distances, and in the end I take the root of that. So that's that's a very standard measure of how well two things fit, which we often use in structural biology. It's got loads of drawbacks, but it's still the most used method, so it's good to know it. Some people do represent the binding site just by amino acids. I told you there are some drawbacks to it, but there's also the advantage that it's really fast because the number of amino acids is not too high. And you have the typical substitution tables for amino acids, so you can deal with that. So this is one similarity measurement, and then you can really go down to physical-chemical properties and score by that. So this is the variations you have in the different methods. Now the example I'm talking about is called Knowledgebase. And what is done here is you take the binding site and you first calculate a surface inside the binding site, like where is the electron shell, more or less. And then for every surface point you look what kind of interaction partners are in the vicinity. And for example, for a CO, you know, okay, it could make H bonds in this area. And then you, yeah,
sorry. Is this the same as looking for potential interaction partners or potential?
Where the same kinds of methods are applied when you do docking.
Yeah, we talked about them before. I was
trying
to know.
So at the moment we're looking for how can you compare binding sites, but the ideas behind that are always the same when you look for potential interaction partners, etc. So what? Yeah, they. So now you look. Okay, I have potential interaction partners around here, and I overlap that with the surface, and now I mark the surface where it overlaps with. Here I could have a hydrogen bond interaction partner. Okay, and then I approximate those surface points by in the vicinity also by one hypothetical interaction point. So they deal with both. With the surface area and with those pseudocenters. And the types of pseudocenters they deal with are hydrogen bond donor. That means it has a proton that could be given to something else. Hydrogen bond acceptor, that is, I want to have a proton. Some things can also be both. For example, an OH can be both. It can be a hydrogen bond donor and a hydrogen bond acceptor. Yeah.
Could you repeat again the whole thing? So first you said that we calculate the surface, then?
Yeah, I have blended out too much of the text. So let's go back in the right order. So you find the cavity with Lixite. This is what you see here. Then you look at the residues that are close to the surface. Yeah, that are like surround this cavity, and you start to classify what kind of things, what kind of interaction partners do I find in there, and I can find those kind of interaction partners. Donor, acceptor, things that can be both. Aliphatic means it makes only the hydrophobic interactions, where only the shells get close, nothing else. And then pi, that means you have those delocalized electrons, and you like to stack them.
Covalent bonds?
No, covalent. So in protein-ligand interactions, covalent bonds are nothing we are really interested in because it's a completely different kind of binding. So it's a lot higher binding energies, and there are some drugs that interact that way. They're called suicide drugs because they can just bind once and then it's done. But in the usual thing, you have only binding via hydrogen bonds and ionic interactions, etc. And of course, some residues can have several of these. So one residue can, for example, have an aliphatic interaction. If it's got a longer chain and an OH, then it can also be a hydrogen bond donor and acceptor. Okay, now we have gone through and looked for each thing. What kind of interactions can the residue make? And then I can also, I can represent all these atoms by those pseudocenters. And in addition, we map the interactions that are possible to the surface. So we have two representations. We have the pseudocenters and the surface. Okay, and then something that will appear again and again in this lecture is that we create a graph. The nodes in the graph are the pseudocenters, and the edges in the graph are the distances between pseudocenters. Now I have a graph representation for one binding site. And if two pseudocenters are close, then I make an edge between them and say, okay, and this is the distance between those two.
Search for clusters?
Right. Okay, and then if I want to superimpose two, I make an associated graph, which consists of the two binding sites I try to compare. And there I have a node where I take one node from protein A and one node from protein B, and look whether they are the same type. So if both of them are an acceptor, then this gives a new node in my associated graph. If they're completely different, I say, okay, I'll take the next pair and see whether this can be a node. Okay? Then two nodes in the associated graph have an edge. If the individual, so if the one node in my associated graph is a pair of nodes from the individual graphs. The other node is a pair of nodes from the individual graph. Okay? So both these nodes have in their original things a certain distance. Now I look at the distance of my two interaction sites in protein A and the distance of my two interaction sites in protein B. Are they similar? If yes, then I can make an edge in my associated graph.
Which connects all these?
Which connects my new node in the associated. Sorry?
It makes it like a group?
Yeah, something like that. So I've got protein A with 1 and protein B with 2, and they have a distance, and they would be connected by an edge. I have protein B with atom X and Y, and I said, okay, these were the same type, therefore I made a node X. Maybe I should take another question. So my associated graph would have X1 and 2y, and now I look, is this distance similar to that distance? And if yes, I say, okay, these two nodes are connected. Because then I know that they are compatible in type and they are compatible in distance. And after I've gone through all those possible pairs, I can then, in my associated graph, look for a clique that is completely connected, and then I know which points I can really superimpose. And this occurs again and again because it was one idea that the computational chemists had, and then they reuse it everywhere. Okay, and so in this case, we only look at cases where this distance is smaller than 12 angstrom, because otherwise it's not really relevant. And the distance difference needs to be smaller than 2 angstrom, because otherwise it's not really a comparable distance. Okay, and then you go through that one and find the maximal common subgraph, and this is the algorithm used there. And in the end, yeah, you get the similar arrangement of pseudocenters. So that was like, how can I find whether I can superimpose them and how to superimpose them? Now I need to do the scoring. I need to look, is this a really good match? And the scoring is based on looking at the surfaces I talked about. So I look for surface patches where a lot of the surface can be superimposed. And the thing that needs to be kept in mind while doing that is that my surfaces look the same, even if one is convex and the other one is concave. So that was the difficulty they had to deal with. Okay, so this is what my colleague Stephan Schmid developed. And afterwards he looked, can I indeed find this kind of gem I want to find, which I showed you in the beginning. So he searched CAF-base with one of the structures, this kinase. He searched against all of the structures that are contained in the database. He threw out everything that has got a high sequence identity, because that's trivial and not interesting. The first 8 cavities he found have a similar fold, so they're not really similar in sequence, which is easy to throw out. But then if you look at a structure database, you see, okay, this is a similar fold, this is probably related. But cavity 9 was actually the protein I showed you in the beginning, which is completely unrelated. And he said, oh yay, my method works. The problem is that searching through the whole database takes on the order of days for one protein. So there's nothing you can really do in a high throughput thing. But then his PhD was finished, and and I mean, that was in 2000 well, the paper is newer, but the PhD was finished around 2000, and then he went to other things. I don't know whether they really try to make this faster. I haven't seen any follow-up since. So a completely different group came along and said, okay, we'll also try to do this job. And they can actually really do high throughput, which is why I'm showing you this. They take the C-alpha atoms of the amino acids. So I think they're probably rather biologists and not chemists, as we were. So they don't really look for, we went to real trouble to say, okay, we want to get exactly the interaction points and things like that. And they say, well, that's too much detail, you can't do that high throughput, so we'll just take amino acid types. Okay, they take the C-alpha atom. Do you know where the C-alpha is, by the way? Because we mention it so often, and you should ask. If anywhere in the lecture we mention words that we mention often, and you don't know, you should ask. We're so few people; it's really possible to just answer that. Okay, I showed you the amino acids, and a general amino acid has got a C in here. It's got this carboxylate group, which usually when it's in a chain, you take away this H and then it goes on with the next part of the chain. You've got NH2, and again, usually one of the H's is gone, and you go on here in the chain. And then attached you have a 1H, and you've got the residue. And this thing in the middle, where everything is attached to, is the C alpha. That's kind of the core of the amino acid, and if we just want to have one simple representation of where does this amino acid sit, we only take the C alpha. Okay, so they just take the points of the C alpha atoms, and then they do something called Delaunay tessellation, and I was really, when I first read that, what the hell is it? And I found out that it's basically you place a tetrahedron across around the C alpha, and then you fill the space with that. So basically you represent your protein by a set of tetrahedra. Yeah. Then they say, okay, we look at two different boundaries. Because this helps you find the binding site. So they take an outer boundary, which connects kind of everything, and they take an inner boundary, and then when you have the difference between them, you can see where a binding site is. And every C alpha atom is characterized by a vector, which has the distance and the directions of the boundary as a property attached. And this is what they call the geometric potential. So they can this is a way of keeping track of where things point and where the inside is and things like that. And also they just take the standard bioinformatics substitution matrix. And again, they make a graph where each C alpha is a node, and you make an edge if the tetrahedra touch. And the alignment is again the associated graph. So if you look for, does my one node in A and one node in B have a similar geometric potential? That is the distance to the surface similar, and is the direction of the surface similar? You give this thing a weight, which is basically you look in the substitution matrix. Is it likely to substitute the two amino acids for each other, because then you assume that they can make the same kind of interaction. So an edge is if the two member nodes, like here, if the two member nodes in A and B are individually connected, so they need to be close to each other. If the distance difference is again below 2 angstrom, and if the surface normal distance is below 30 degrees, so the direction needs to be comparable.
So are these thresholds defined based on what kind of interactions we are looking at? For example, if we consider...
I'm not sure how exactly they did it. So I guess it's probably kind of intuition, where you say, yeah, if the distance is too big, then it won't make similar interactions possible. And usually you also do that by fine-tuning. So you make your method, and then you see, can it do what I want it to do, and then you fine-tune the parameters.
Because the hydrogen bomb had a distance of...
Yeah, but in that case, we only represent the whole thing by the point of the C alpha. So we are not really coding for the hydrogen bond at all. So, as I said, this is pretty different between the two methods, but I have to admit that this one is faster. And it does its job, so... Okay, and in this case, they say they want to, the difference from the other one is there we just had to find a connected subgraph, and here we also have to look for weights, because every thing has got directly, so they don't do the scoring afterwards, they kind of do it while finding the subgraph. Yeah, and the weight is calculated by the distance, the normal vector angle, and the substitution matrix. And what they did is, because they were fast enough to do this over the whole database, they looked at the statistical distribution of their scores. So they took one or several proteins and compared them against the whole database, and then they look at, okay, what's the statistical distribution of what kind of scores can I get? And then they analyzed that and they said, okay, this kind of score is significant. So in the other case, it was not so easy to tease out what is a good score and what not, and especially if they basically only took one pocket and then looked for what kind of other pockets are similar. Here they want to do a more wholesale thing and take several pockets and look for everything and then see whether the different scores are comparable, what is likely to find by chance, etc. So what they did in order to find the background score distribution was to compare completely unrelated structures with some random sequences, and in the end they were able to give some probability scores. And they also cite some nice application examples. For example, they wanted to find out why some estrogen receptor modulators have side effects. Estrogen receptor modulators is something that you need, for example, to treat breast cancer. So if people have breast cancer, usually what the target is estrogen receptor, and you try to block that. But of course, any drug always also does something else, and they wanted to understand what else does it do. So they said, okay, probably the off targets that are hit will be somehow similar to my real target, and I'm going to see what is it similar to. So they mapped all the sequences from the switchboard database to structures, because not every sequence you might be interested in has a structure. Only about 5% of the sequences in switchboard have really a determined experimental structure. So they first had to map that somehow to structures. They limited, in order to not to have to search too many things, they had a definition for what is druggable. What kind of protein can I bind to in the first place? So that is kind of redundancy reduction or search space reduction. Then they searched with their algorithm, and on a pretty high rank they found a protein called SERCA, which in the end is a C-alpha channel ATPase. That is something that transmits signals, and one of the signals it transmits is, for example, for muscle movement. And then it was easier to explain the side effects, which is, for example, cardiac abnormalities. So because then you get the wrong signal. Yeah. So they had, and then they tested further and found that this really, the ligands that bind to ER alpha also bind to that one. Yeah.
Did we mention soy pot?
Sorry, that's the abbreviation for the method. I think it's mentioned somewhere on this slide, but this is the method I just presented with the tessellation and everything. Okay. And here you see a comparison between the two binding sites, one from the ER alpha and the other one from SERCA. And overall, the form looks pretty similar. The electrostatic potential looks at least comparable. So you've got the same kind of charge distribution. So it does make sense in a way. In order to validate this, they also did the inverse search and took the sequence they found, or the protein they found, and searched it also against the whole database and looked for, do I find this ER alpha and height score? And here you see score distributions for. They took different parts of the binding site and looked for that one. And in both cases, they found ER alpha at a pretty high score. So they said, okay, this is probably a significant result. Okay, so this was one example of how they applied it and said, yeah, yay, we've done something with our method. And the other application example is you always need new drugs, especially for diseases like tuberculosis, because there the bug starts to get resistant to the known drugs. And also, yeah, and they said, okay, instead of starting new from scratch, I want to take a known drug which has been optimized against side effects, etc. already, and I just want to use that against tuberculosis. And then the idea was, okay, how do I choose which drug to start with? In order to treat tuberculosis, it's good to have something that is able to bind adenine, because that is a part of the binding site that you want to block in the tuberculosis bug. So they looked for things that can bind adenine using their method, and they found a certain type of proteins. And now they superimposed the two structures, the target they want to hit in tuberculosis and the one they found. And again, they can find that so in green you see the protein that was found in, should be blue. I think somewhere the color scheme changed in PowerPoint. In blue you see the protein target you wanted to have. Both of them have got so-called cofactors, which are sitting in there and used for the function of the protein, and the two cofactors align pretty well. And the inhibitor of the found protein aligns pretty well with how a ligand could bind into the target protein. And so they then tried out this compound, and indeed it does block tuberculosis growth. Okay, so that was comparing binding sites. Do you want to have a break, by the way, or just go through? Because if we make a break, then this would be the point. Okay, good. So now we compare binding sites. Now we're going to come to the point. At this point, it would of course be nice to first in the computer try and see whether this molecule fits. And how do I do that? How do I dock? If you want to dock, then there are again a few things you need to be able to do. One thing is to tell, is my docking pose sensible? The geometry I just generated, is that sensible? That's what we call scoring. Another thing is we of course have to first find this pose, this way the ligand could find in there. This is called positioning, and I'm going to talk about these two things, how you do that. I'm going to show a few example programs, and then in the end I'm going to tell you a bit about how well do docking programs do nowadays. And in the end show an example of how you can use it. Okay, I think I'm going to skip that one. Some general hints on how to develop a good drug. It always helps to have some lipophilic contacts. So the point is that inside the binding pocket you have some parts that are hydrophobic, and around these the water is more ordered than in other parts of the molecule, the binding site. If you throw them out, you have an entropy gain. So if you have a ligand and a binding pocket that have a lipophilic interaction, that gives you an entropy gain. It's usually not so easy to say what hydrogen bonds do, whether they help or not. And the point is hydrogen bonds are good, they make a good interaction, but the protein and the ligand can also make hydrogen bonds with water. So in the end, this can cancel out. On the other end, it helps to anchor the molecule somehow in the binding site. And the general rule is if you have polar protein atoms, you mustn't bury them, so they shouldn't have a hydrophobic interaction partner. That doesn't make sense. So if the binding pocket presents an interaction partner, you have to use it. And the other thing is, if your ligand is rigid, then it's usually stronger because it doesn't lose entropy on binding. Okay, so now we want to be able to calculate whether the pose we have thought of is going to be good and whether the binding is going to be good. What we have to be able to do is to distinguish likely and unlikely conformations. And we also have to be able to distinguish good and bad binders. These are two different things. In one case, we just compare one ligand with different docking solutions and want to know which one is the most likely one. And in the other one, in the other case, we want to compare two ligands and say which one is going to bind better. And our general assumption when we do docking is that the bound state does have a minimum. It could also be, I mean, one thing you have to always keep in mind, because in bioinformatics we always show you pictures of proteins and it looks like this is the protein and it looks like that. But that would be like making a photo of you and saying, okay, you sit there. You don't do that always. And the proteins also move. And also a ligand that is bound somewhere can move a little. Usually it will have a kind of minimum there, but it can also move. So this is something you have to keep in mind. But the general assumption we make whenever we do this is that there is a minimum and we just have to find it. Okay. So what do we have to consider if we want to calculate the energy? Well, one thing is that not only do these two things make interactions, but the protein and the ligand have internal energies. And this is just to illustrate in a very simple case. If you have a butane molecule, so 4 Cs, and these things are supposed to be H's, and you have one rotatable bond here in the middle, then there is an energy difference between the different conformations. So if the big substituent of those C's points in the same direction, then they can have a bit of a clash, and that has a more disfavorable energy than if they're turned the other way around. This is something you have to keep in mind when you put a ligand somewhere, that it also internally has some energies. So typical force fields for calculating these energies approximate everything by springs. So we assume that the atoms can move relative to each other, and we approximate that by a spring. We assume that the angle can change. We approximate that by a spring. And finally, the things can rotate. And we don't really approximate it by a spring, but this is kind of the typical way to represent it. And always we have some standard bond length, which has got the minimum energy, standard angle, and standard torsion angle, and we have the force constant for the springs. And these need to be got from somewhere. And the collection of these parameters is called a force field. So for every type of bond, like CH, CC, CO, whatever, you've got a standard bond length and you've got a force constant. And the collection of all that is called a force field. What are properties of force fields? Well, what you can calculate with that is only the enthalpy, not the free enthalpy. Last time I talked a bit about thermodynamics and said the difference between enthalpy and free enthalpy is, does it contain the entropy change or not? And the force field like this cannot say anything about entropy. This is something to keep in mind when you deal with this kind of function. The collections of parameters that are mostly used are called charm and amber. It's just, for you, just a term. And the only relevance it has for you is if you come across the term charm and amber in the future, you know, that's what they're talking about, this collection of parameters. Okay? And this is what these force fields look like. Again, I don't expect you to be able to reproduce the formulas. This is just to give you an idea of what this looks like. Because they are similar in many respects, but they also have dissimilarities, and it's good to have a feeling for how can they be similar and dissimilar. So you see in both cases, they have the bond term, they have the angle term, they have I think the torsion is represented differently, and so the dihedral and torsion is more or less the same. They also have some improper torsions, which is something specific, and I don't quite remember. They have non-bonded interactions. So the standard, you might recognize the van der Waals we talked about, Lennard-Jones terms we talked about last time, where you have the repulsive and the attractive forces between the electron shells. You've got the Coulomb term. Okay. So now you can, when you see that, you can kind of understand what this is talking about. Okay, so these are force fields, and they try to approximate things from ground up. So the way where they get the parameters from is that they take loads of small molecules, observe them by spectra in gas phase, etc. Look for how do the bonds wiggle, etc. Then they try to simulate that with molecular dynamics and optimize the parameters for that. And in the end, this collection of parameters is then used for other stuff. Okay. So in a way, these things are empirical. You know, they're not calculated by quantum mechanics or anything. They also come from measurements. This is another kind of scoring function which we call empirical, but which is kind of only another type of empirical. They don't start from the internal energies in the way I just told you about, but they start by trying to approximate binding energies that are observed. And one example is the scoring function from Autodoc. Another one is CAM-score. So again, as before, we've got the van der Waals term. We've got the Coulomb term for interactions, but then you have a specific H-bond term, and you just count the number of torsions and have parameters for how the torsions change the energy, and you've got a solvation term which just looks at, okay, how much of what surface of the ligand, what surface of the binding site is solvated or desolvated, and I have a term for that one. Okay, and the last type of scoring function we have is knowledge-based scoring function, where we observe how often we find certain kinds of distances in crystals, and then approximate from there. And the way we do that is that we go back to the Boltzmann distribution. I don't know how many of you have heard about that before. Probably not. Okay. So this comes from statistical thermodynamics, so quantum mechanics and things like that. And what you can see is that the probability of finding a system in a certain state is related to the energy of that state. The higher the energy you need to make that state, the less probable it is. And you can ignore the rest. So the main thing you should know is it's related to the energy. Probability of a state is related to its energy. You can then use that to compare different states, and in the end you see that you can calculate an energy by comparing the probabilities. Okay, and now the tricky thing is to find what do I compare against. And for example, also one of my colleagues from my PhD developed a scoring function based on this principle, and he looked at lots of different interactions, that like different C's that are aromatic, different C's that are surrounded by, for example, H's, etc. And then he looked at the distribution. How often do I find a C in that distance? How often do I find two C's of that type in that distance? And he also defined a reference state, and then he could always compare those against the reference state and make an energy from that. And in order to be able to somehow capture solvent effects, he made the same kind of statistics for desolvated surface area. And in the end, you get a potential like that, and you have to optimize it. And in the end, this drug score was able to pretty well reproduce or pretty well find good poses amongst docking solutions. Okay, so now we have different ways of finding a good solution or of scoring. And the other thing is that we need to produce the thing we want to score in the first place. Okay, so you have a lot of degrees of freedom because your two molecules can move in space, they can rotate, they have rotatable bonds within them. And you need to optimize that. Traditionally, docking, so the first docking programs completely ignored rotatable bonds in the protein. So that's too much. Nowadays, at least you rotate the amino acids a little. And in the beginning, usually you also assumed that you have found a good confirmation of the ligand, and you just dock that. This has also changed over time. So the first docking method just first found a good confirmation of the ligand and then docked that. The problem is that something that has a very low energy for the ligand doesn't necessarily also really fit well into the binding pocket. So even if the energy for the internal energy of the ligand is a bit higher, it could be compensated by good interactions. Therefore, that's a bad thing to do. But that was in the beginning because they just wanted to reduce the search space. Okay, typical algorithms for searching this thing is matching the shapes, constructing the ligand incrementally inside the pocket. So you first have an anchor and then you extend that. Genetic algorithms are applied often, so you have different, you encode the torsion, the movement, and also internal things into a long string. And then you combine those strings amongst each other and always score and have a way to find the optimum there. Another way is simulated annealing, and Monte Carlo methods. are not going to go into details there. And you see that some docking programs also offer different options, so the user can choose which kind of optimization he wants to have. Okay, let's look at a few docking methods in a bit more detail. One is dock, and there the basic idea is you take the binding site and fill it with spheres. Because you asked, or no, one of you asked last time whether, I don't think it was another lecture, whether you can also take spheres. Yeah, you can also, instead of filling the binding site with a grid and calculating stuff for that, you can also see what are the biggest spheres I can take in there, put in there. You also fill the ligand with spheres, and then you can try to match the spheres onto each other. And the spheres are, as with the superimposing two different binding sites, you can mark the spheres with properties and then try to find matches between compatible properties. Okay. Yeah, as I said, it's basically the same as what I showed here for matching binding sites. You have ligand atoms and you have protein spheres. You have compatible matches between them. You look whether they're distance compatible, and then you search this matching graph. Okay. And later, so first dock was dealing with completely rigid protein pockets, completely rigid ligands, and later they introduced ligand flexibility, and there they first started to deal with anchors in the ligand, which have good interaction partners, and then grow from there. And if they, while they're still searching the whole space, they have a pretty crude scoring function where they just look for, do I have any bumps? Because if the two things overlap, the protein and the ligand, then this is not going to work. And then later, when they have extended the ligand further, they start to look for real energies. And this is to save computation time.
And how do they decide when to stop? Like iterating?
I'd have to look up the details again.
It says complete. So the proof.
Yeah, well, they build up the ligand. So you're complete when you've built up the ligand completely. Okay. So in Autodoc, the receptor is represented as a grid potential. As I introduced in the first lecture, you can put the grid inside the binding site and pre-calculate potential interaction energies for every point in the grid. And these interaction energies come from an empirical scoring function. It starts with some random location and orientation, and then from there it explores what kind of rotations and conformation changes, etc. you can make. And here the different kinds of optimization method that I showed just before apply, and you can just choose. You can take simulated annealing, where you change a little, then you look how much is my energy difference, and the probability of accepting a different pose depends on the energy difference. And so the lower the energy is, the higher is the probability to accept it, but you can also go over barriers because they have a limited probability to also go up in the energy. The genetic algorithm, as I just mentioned, you encode all the movements by a bit string, you recombine that and let that run for a while, and you take a certain number of descendants depending on the energy again, which you calculate. And yeah, the Lamarckian genetic algorithm, I always find that funny because, I don't know how much biology you know, but there is Darwinian evolution and Lamarckian evolution. Do you know what Lamarck thought, how we evolve? So in Darwinian evolution, we kind of know that our genes encode for the phenotype and just the phenotype that is fit is selected. Lamarck thought that we can also evolve by changing our phenotype, and then the descendants inherit the phenotype. So, for example, a giraffe would grow a long neck because it always tries to get the high branches, and the finished giraffe still grows, and then the children of this giraffe also have longer necks. Okay? We know that this is on the large scale not true. Meanwhile, we know that it might be a little true on the molecular scale. And the funny thing is, in computers, you can of course do that, because when you have encoded something, you can change that a little, optimize, and then let the children inherit the change state instead of the gene you had in the beginning. Okay, so that was Doc and Autodoc. FlexX is by the same people, or kind of the same people as the CAF-base idea. So we go back to the point where we look at types of interactions, and we classify interaction sites. But first look at ligand flexibility. They looked at how probable is it to find certain torsion angles in databases. Then from that you can approximate an energy function. Then they looked in a lot of detail at the different interactions you can have. Like, they make a difference between an acid group, for example, that has a protein attached and that doesn't have a protein attached, and looked at, okay, what are ideal distances, what are ideal angles, etc. So they had a very detailed representation of interactions. And so they were able to really well model hydrogen bonds. Have an empirical scoring function, and so that was the part that the chemists did. And then they talked to computer scientists, how can we make this efficient, and they found the thing that they first place a base fragment, and which has many interaction sites, because that defines the starting point really well, and then they grow from there. In order to place the base fragment, they again do some kind of matching between the interaction sites in the ligand and the interaction sites on the receptor, which basically goes down to that kind of algorithm again. They score each base placement, and from there they do a tree search for how can we extend from here. And in order to be efficient in the tree search, they take the directional interactions first, because they're most defined. So if you have an OH, an H bond, this has to have a certain conformation, otherwise it doesn't work. Whereas if you have a hydrophobic interaction, it's more flexible about the exact geometry. Okay, so now you have a few ideas of how you can do that. And again, the main point is to get you a feeling for how you can solve the problem. The details are sometimes a bit arbitrary, which exactly do you choose. But now if you're given the task, please doc this, you have to choose a program. And of course, people also try to compare, okay, which program is the best? And I'm going to show you a bit about this evaluation of which docking programs are the best. How would you evaluate it? What can you do? Well, one thing you can do is to look at, I have 3D structures where I have a ligand inside, and I can see, can I reproduce that? Can my docking program take out the ligand and then find the same kind of pose again? The other thing is I often want to do is to find a good binder. So if I screen against the whole database, I want to find those that are good. So I take experimental values and see, can I find those back? So this is finding back is called enrichment. So I look how many or what's the percentage of hits I would have selected based on my docking program compared to what's the percentage of all of the database. Okay, so here's an example for a redocking I did in order to try out a program which I used. And in green you see the reference ligand, and in yellow you see the best docking result, but that wasn't found on the first rank. And in blue you see the thing that was found on first rank. And to just give you an idea, the RMSD of the best docking solution to the real one in the crystal structure was between 1 and 2 angstrom. Just to have, because later I'm going to talk about RMSD values, and to just give you an impression of how good is the overlap, I'm showing you this example. So this is between 1 and 2 angstrom, closer to 1, and that one was between 2 and 3 angstrom. So you see, you can have quite a bit of a difference if you're in the between 2 and 3 range. Okay, now if you take loads of different docking methods and loads of different ways of generating the ligand conformation. So one is you just take you take the ligand as rigid, but you take it in exactly the conformation that it has in the experimental structure. And in the other cases, they tried out other ways of generating the conformation. Then you see, and here we see the average RMSD of the solutions you find, and here we see what percentage of ligands did they get in at all. Because you don't always find a solution. Sometimes the docking program says, sorry, this doesn't fit. Okay, so what you want to have in a docking program is to have a low average RMSD and a high percentage of solutions. Okay? So you see in that evaluation, Gold was doing pretty good, and something called EHITS was doing pretty good. FlexX was not doing so good, but there I have to say, well, they didn't let FlexX try out its own conformations, but they fed it conformations. That's a problem you usually have when you have different comparisons of tools. They try to be fair, but then in a way they're also unfair again. Therefore, it's very hard to say something about it. But you get a general idea. You see, okay, this is the range of RMSDs we get into, and this is the range of solutions we find. Now this was for the top scorers. This, if you only look at the first solution, and this is if you say, okay, I don't really care where in the hit list the best solution comes, I just want to see whether it finds the best solution anyway. And then you see the RMSD gets lower. Yeah?
What's on the x-axis?
Here you see, oh sorry, it's swallowed again the mean RMSD. So as here you have so you have lots of different things that have been docked, and you just calculate how much is the RMSD in general. And you see that the values get lower. So on later places in the hit list we do find better solutions, which means that the scoring function is not optimal. It doesn't always it's not always able to pick the best hit that a docking program gave you. And I have to tell you from real applications, computational chemists who help develop drugs in pharma companies always look at like 10, 20 solutions and not just one. At least if they're really working on the details of something. If you're scanning a whole database of 1 million compounds, you don't. Okay, and I don't know how they calculated the percentage of successfully docked pairs. I can't quite remember. The point is it gets more distributed towards lower values. Now, this was, are we able to reproduce a good pose? Well, it's okay. You have to keep in mind, also look at not only the best scored ones. Are we able to predict how different ligands bind relative to each other? And do the docking scores correlate with the binding affinities we can measure? And you can see here the different correlation coefficients are not really high. So at least, I mean, we're able to say the trend, at least the binding, the correlation coefficients aren't negative. That's a good thing, but they're not really close to 1. That's also something to bear in mind. However, I have to say, also as a side note, if you have different groups measure the same binding affinity, you're also not going to get a 1 here. Because measuring binding affinities always depends a lot on the exact details of the experiment. And you can, so in the group where I did my PhD, we had some people who did this experimentally and also compare it against other groups. And then because they had different results when they did it in different ways, they also looked in the literature, what do I find there? And they said you can easily have a difference of a factor 10 or even 100 between different groups, because it depends so much on the exact conditions. So that is to keep in mind when you try to compare against experiment. You really have to say, okay, which experiment do I really try to compare against? The final thing was enrichment in screening. So I take a whole database of ligands and I try to find back those where I know that they are active. And the ideal enrichment in this is always shown here, and you see how close different kinds of methods get on different kinds of targets. And you see it depends a lot on what protein you're dealing with, and you can't always say that this method is better than that one. But in general, at least you do get an enrichment. There's also something, you know, instead of so if you select, or what happens in a pharma company is they take this whole thing in silico, they take a database of ligands in silico, then they look for what could be a good hit, and from those they take a bunch and then test them experimentally. So it does save effort to do this first. Okay, one example for where docking has been used successfully is you have the problem that you want to develop a drug against HIV. The problem you have there is that HIV mutates a lot and makes the inhibitors inactive. So what you want to do is to find something that is going to bind in the wild type and also in some mutants. So it's insensitive to the mutation. And in order to find those molecules, they docked a library of 13 million molecules against the wild type and some mutants. They then looked, okay, which are the top scorers in all the different mutants, and for those they took a more careful docking program, more careful scoring function, and in the end they visually inspected 1,500 structures and chose 9 structures where they thought, this could work. And actually in 3 of them they found a micromolar binding constant, which for a pharma is okay. It's not really a good drug, has a binding constant in the nanomolar, so 10 to the 3 less concentration, but it's a good starting point. And so they developed from there on. Okay, and I had shown you before this thing about the estrogen receptor, where they looked with their program to find similar sites. In addition to just finding that, They also took the ligands that bind to ER alpha and looked for how strongly do they bind to this other protein by docking. And then look, do these things correlate? This was another way to validate whether the two binding sites are really similar. So this is another application of where you can use docking. Okay, to sum up the whole lecture, we can dock. The performance is that we can get to correct structure in about 70% of the test cases. The prediction of binding affinity is very difficult. We can rank different geometries. Okay. We can rank different ligands. Well, we get correlations. And to really estimate the exact value, we don't really get that. And the challenges we still face in protein-ligand docking is protein flexibility, because it extends the search space too much, and yeah, improving the reliability. But still, docking is better than random guessing. So it does help, it's applied, and therefore it's also an active research topic. Okay, so that's it for today. Any questions?